Today I will be discussing transcription in prokaryotes. Transcription is the process of replicated DNA being transcribed into an mRNA sequence. In this video, I will go through the process of initiation, elongation, and termination that happens in prokaryotic transcription. Starting with initiation, I will replicate the promoter region of the DNA sequence, which is where the RNA polymerase will bind to and continue into the elongation phase transcription. First, we have the negative 35 region, which is one of the sites that the sigma subunit will bind to, along with negative 10 region, which is another binding site for the sigma subunit of RNA polymerase. Both these regions are consensus sequences that describe the nucleotides present and are located upstream prior to the start of transcription. We can hypothesize that there is a 5-base pair insertion between negative 35 and negative 10 region of primary sequence and the gene that codes for amino acid phenylalanine. It would likely cause the sigma subunit to not attach properly to the negative 35 and negative 10 region required for transcription to occur. This simply binding error could lead to an inhibition enzyme phenylalanine being produced from this gene sequence, which would allow for the phenylalanine to carry out its signaling functions to other molecules in this prokaryote. Moving to the downstream region of our DNA sequence, this region indicates sequences after the start of transcription, which includes the +1 site. This site is where transcription will initially begin as RNA polymerase moves down the sequence to form the mRNA transcript. Now we know what starts the initiation process of transcription, we can discuss RNA polymerase that aids in the process of initiation and elongation of transcription. RNA polymerase is an enzyme that synthesizes the mRNA strand from transcription. It is made up of a core enzyme along with the sigma subunit that creates a whole enzyme, needed for RNA polymerase to bind to the protein regions. An analogy for RNA polymerase is that our core enzyme represents a car with no engine. Without an engine, our car will not be able to move from point A to point B. Now our sigma factor is the engine. With our sigma factor and our core enzyme, we have all the complete parts attached to point A sequence and continue down to our point B. Now that we have our initiation sequence set up, we can go ahead and move on to how our mRNA transcript is made through elongation. First, I've labeled that transcription in our example we move from left to right, where I've labeled the 5' and 3' regions, keeping in mind that we add nucleotides to 3' to 5' and read the RNA transcript from 5' to 3'. Additionally, I have labeled the bottom line as our template strand, moving from the 3' to 5', and our top line as the coding strand. Once RNA polymerase attaches to consistent sequences of the promoter region, it will create a transcription bubble that will allow for the RNA transcript to be created and read 5' to 3' from the template strand. Once 9 to 12 nucleotides are added to form RNA, the sigma factor RNA polymerase will dissociate. Moving into our bubble, our RNA is helped synthesized by RNTP, which is the building block for RNA synthesis and serves as a chemical energy storage system for this process. RNA polymerase will move down our sequence into 3' to 5', the template strand, synthesizing our RNA strand in the 5' to 3' sequence. Finishing our elongation phase of transcription, new nucleotides are added to our RNA transcript, where phosphodiester bonds seal these nucleotides together like superglue for the newly synthesized RNA transcript. Termination occurs when RNA polymerase moves onto a terminal region of a DNA sequence, signaling transcription to cease. The two types of termination sequences I will be discussing briefly is row-independent and row-dependent. Row-independent termination occurs when the RNA reaches a point in the gene sequence where there are inverted repeats of both guanine and cytosine configurations. These repeats cause the RNA chain to form a stem loop, otherwise known as a hairpin. This hairpin structure ends with a tail containing uracil, which matches up to an adenine tail in the DNA template strand. The interaction of this loop is very weak and will thus dissociate and allow for the RNA chain to be released. Row-dependent termination occurs where there is a row factor protein binding site, otherwise known as a RUT site. When this protein binds, it continues up the RNA transcript towards the 3' end of the transcript into RNA polymerase. This causes RNA polymerase to pause at termination hairpin and allows for the row protein to use helicase activity, which causes the RNA to unwind and cause transcription termination.